Hello, this is Tom Repass of Canyon Rim Honeybees in western South Dakota. This presentation is about Varroa mites, biology, monitoring, and management. In this presentation, I will review what Varroa mites are, why you should monitor and treat for mites, how do you monitor, what are the current treatment options, and when you should consider treating. When I first started keeping bees back in the 1980s, we did not have to worry about Varroa mites, but they have become one of the greatest challenges to keeping bees successfully. That's something that we always have to worry about at some level. What are Varroa mites? They're a parasite that feeds on both the adult bee as well as the larva and the pupa. They were originally a pest of Apis cerana, the Asian honeybee, and they had co-evolved so that they did not cause any great deal of harm to the original host. However, when they crossed over to the European honeybee, those bees had no natural defenses, and it decimated the beekeeping in every place where they arrived, Europe and then eventually the United States. They're everywhere except Australia, although this past year there has been Varroa found in Australia, and they're trying very hard to keep it from becoming more widespread. Besides the fact that they parasitize and feed on the larvae and adult bees, they also are a vector for diseases such as viruses. Every honeybee colony in the US and Canada has mites today, or they will have them very soon. And one problem that I see, many new beekeepers go into beekeeping and they have wishful thinking, and they hope that, well, I didn't see any mites on my bees and I didn't treat this year and they did fine, so I don't have to worry about them. And that's really wishful thinking. You always have to be thinking ahead about what you're going to do. And it might not always mean that you're going to be using treatments or chemicals. There's other management techniques you can help to maybe not treat or cure the hive of the mites, but at least to keep the mites under control. And again, they're located just about everywhere that there are honeybees, and most recently they've been found in Australia as well. In order to manage mites successfully, it's important to understand their behavior and their reproduction. So the adult female mite enters a larval cell right as the prepupa spins the cocoon and right before it's capped. About 60 hours after entering that cell, she will lay her first egg, which will always be a male, and then about every 24 hours after that, she lays a female egg every 24 hours. Her son, the male, develops and then mates with his sisters as they mature. Incest is a normal thing among mite reproduction. When the adult bee emerges, about 1.4 daughter mites will be produced. However, if she entered, and that is a worker, if she entered a drone cell, then she may produce 3 times as many daughter mites. The reason is because drones take longer to mature than do worker bees, and so that extra few days of time allows her to produce more daughter mites, and that's why often they will prefer to enter drone brood over worker brood. This is a set of videos that I found online, and they're extremely fascinating. And so this is a pupa that's been removed. You can see the mother mite feeding there on the lower left, and that tends to be where the feeding site always is, and they don't know exactly why. The mother mite will create a feeding site, and that's where all the daughter mites will also feed. And you can see some of the other mites crawling around within that cell, and then you can see some of the white excrement from the mites. You can see a couple of eggs there on the side of the cell. And here is a little video of the male mite, and he's much smaller than the females. And of course, in the cell, when it's closed in the hive, it is pitch black, so they sense the other mites based on chemical signals and also feel. And here is the male mite mating with one of the sister mites. Now, for many years, we used to think that the Varroa fed on the hemolymph. Hemolymph is kind of like blood in insects. But as Dr. Sam Ramsey discovered, they actually are feeding on the fat body. We used to think that when the mites were hanging out on the adult bees, they were just hanging out. They were what we call phoretic. They weren't doing anything. They weren't causing any harm. But we've learned that they actually are feeding on the adult bee. So they actually are parasitizing. They're not just phoretic. They're not just attached and going along for a ride, but they're feeding while traveling. This is important because those bees can be damaged by mites, and let's say you live up north, the bees that are mite damaged may not survive winter, and that's a common cause of early winter losses. You may go into winter with a very populated hive, they may seem fine, and then the first warm day, say in January, the bees are all dead. Plenty of honey stores, and they all died, and it frequently would be a mite kill in that situation. Now, sometimes new beekeepers say, well, I don't have any mites on my bees. I looked at the bees coming and going at the entrance, and I didn't see any mites. You have to remember, most of the mites are not on the top part of the bee, not on the dorsal surface. Of course, we see lots of photos on the internet because it's very striking, it's very obvious, but less than 1% of the bees will be crawling along on the top part of the bee. So if you do see mites crawling on the top of your bees, you've got a really serious problem, because those are only about 1% of the bees that are on the adult bees, and if at least 80% or more of the mites are actually in the brood, if you do see mites crawling on top of the bees, you basically are at the point of that hive is about ready to collapse. As it would turn out, most of them tend to attach to the left side of the abdomen of the bee. We don't know why. Perhaps it is more easy access to the fat body or some other reason. After I saw this data, I remember all the photos that I've taken of mites feeding on the bee, and yes, most of them were on the left side, and I had never even thought about it. But less than 1% are on the dorsal surface. So why should you monitor and treat for mites at all? Well, the top 10 reasons to lose your colonies in the U.S. Number 1, I would put Varroa mites. Number 2, viruses and disease spread by Varroa mites. The colony dies over the winter because the winter bees were weakened and by the mites and viruses, and they could not survive the whole winter. The colony disappears due to mites and viruses and diseases they spread. There's poor or spotty brood due to parasitic mite syndrome. Oh, sure, bees can die from starvation if you took too much honey or it was a bad year and you didn't feed them. Sure, they can starve. That's a very preventable cause of a colony to die. Sure, the queen can fail in the middle of the winter and not be able to replace itself. Of course, you can have brood diseases. Some places you may be unlucky and have pesticides. But the top causes of colonies to die now are Varroa mites or colonies weakened by Varroa mites and the diseases they spread. Like I said, these are preventable losses, but probably the worst reason for bees to die is beekeeper neglect. Wishful thinking, assuming you don't have a problem, it's going to catch up with you and you will lose hives. So the mites feed on both the larvae and the adult bees, and they weaken the emerging bees, so they may not be able to survive quite as long. They also transmit viruses and activate replication of viruses. And typically the colony will die within only a few years, sometimes even a few months. And this is why someone can buy some new bees, be a new beekeeper, and they did fine the first year and the first winter, but then the second and third years they begin losing colonies if they're not on top of the mites and not managing them appropriately. So in a healthy colony of bees, you want to see a very healthy brood comb, something like this. Not a lot of missed spots, not a lot of open combs, but in a colony that is about ready to collapse from mites, you can see what we call parasitic mite syndrome. The bees are uncapping the brood. Some of the brood has died. Sometimes you can see signs of viruses. This is deformed wing virus right here, this poor little bee. And you can sometimes also see bees dragging pupa out of the colony with mites attached. They're trying to defend themselves against the mite infestation, but the numbers are just too large that they can't succeed. And here's another picture, a close-up of a poor little bee that has the deformed wing virus. I don't know how many times I've been asked over the years, either face-to-face or I get a private message, our hive died. Can you help us figure out why it died? We're so sad. We don't know why our hive died. And I'm like, well, most likely it was mites. Oh, no, it could not have been mites. We didn't see any. Did you do a mite count? Did you treat for mites? We didn't treat because we didn't see any. Here's some photos of our dead bees in the hive. This was actual conversation I had, and these were phones sent to me while I was out in my bee yard taking a break from working hives, sent to me over the phone. And here's pictures of the poor dead bees in their hive. And if you look closer, you'll see that there was a very small cluster, very few bees left. And if you zoom in more, I said, yeah, this colony died from mites. And they're like, what do you mean? And so I circled all the mites that were there amongst the dead bees, zoomed in, and sent it right back to them. These mites are about the size of a pinhead, so unless you look closely, you may not, see them if you don't know what you're looking for. So remember, just because you didn't see any mites on the bees doesn't mean they aren't there. When there's brood present, 80% or more of the mites will be in the brood, and only about 10% or 20% will be on the bees themselves. Now, this can change. As the colony begins shutting down brood production at the end of summer and into fall, you might begin seeing many more mites on the adult bees themselves. But most of them are on the ventral part of the abdomen, like I talked about before. And unless you are looking, you're doing a mite wash, or, like this bee is a photo I took from within my observation beehive where she was crawling on the plexiglass where I could get a really good visual of it. If you didn't see them, that doesn't mean they're not there. This is a slide from Dr. Megan Milbraith, and I share it with all my Varroa presentations. Basically, so many new beekeepers start out this way, wishful thinking, and they lose far too many colonies of bees, and they buy more bees the next year, and then they lose colonies, and then they buy more bees the next year. Eventually, unless they're, independently wealthy and they can just throw money around, they eventually decide, I'm going to do something else. Maybe their spouse is like, you got to do something else. This is getting expensive. Or they begin taking Varroa seriously and take action to keep it from decimating their colonies. So how do you monitor for Varroa mites? There's several ways. You can look at the mite drop out of the entire colony. You can look at the capped brood, scrape off the cappings and look to see if there's mites in there. That comb that is in the photo there on the right, that hive obviously is on the verge of collapse. That's a terrible infestation. Or you can do a mite roll or mite wash on the adult bees, and there's different ways of doing that. So the mite drop is the easiest to perform. You just look at the mites that drop out during a 24-hour period. It doesn't take a lot of time. Mite wash, that takes a little bit more time to perform, but it is a more accurate estimation of colony mite population. So looking at mite drop, say over a 24-hour period, it doesn't tell you that much in regards to what the population is in the hive. All it tells you really is there are mites in that hive. That's about all it can really tell you. There are times of the year where there might not be a lot of mite drop. For example, in the spring when they're building up, all of those mites are trying to go into the capped brood and stay there as much as possible. Whereas in the late summer, fall, not only is the population of mites higher, but there's less brood available for them to go into, and so there may be more mites on the adult bees, and so they're more likely to drop off. We used to think that the screen boards would help with mite control, but it's been shown that they really don't. But they are still useful to look at, how many mites are dropping. Or if I'm treating with mites, I can see how many mites were killed out of that hive. And, I have to admit, I still take great joy in seeing a bunch of dead mites falling after I did a mite treatment. And you have to look closely. Sometimes you won't see very many, and sometimes you'll see a lot. And that's kind of how I look at it. I tend to use it more after a treatment. If I treated a colony and very few mites fell, that's a good thing. Maybe that colony has some mite-resistant traits that I may want to breed for. Another colony may have a huge number of mites, and perhaps that might be a colony that will not be within my breeding program. So another way of looking for mites is uncapping and examining brood. And sometimes this happens, you might have drone brood that's in the comb on the bars of the frames, and as you crack the box and lift it up, you might see some of the mites within the drone brood. This is not a very, sensitive, it's not really telling you how bad the infestation is, but suffice it to say, if you see a lot of mites within the drone brood, that means there's probably a high population of mites in that hive. And if you don't see any, then, maybe that hive, at least for right now, might be okay. Here's some examples of some terrible mite infestations, and these are hives that are about ready to collapse. But there really isn't a good way to know, how does this translate to how many mites are in the hive total. And that's why many of us, including myself, really recommend mite wash. You're using a half cup of bees, which is about 300 bees. I can't stress this enough, do not accidentally include the queen. What I tend to do is I find where the queen is, put her on a comb away from where I'm going to be gathering bees from the brood comb to do the mite wash. The alcohol wash is the one that is most standardized, so that is what I do. And a lot of folks, even me, I don't like having to kill bees in my hive, but I think about it, 300 bees really, in a full-strength colony is a minimal number of bees to be sacrificed, and they're maybe not doing this willingly, but you're sacrificing those bees so you can get information so you can save the whole colony. You think about it, if a bear attacked that hive, you can bet more than 300 bees would willingly sacrifice themselves to defend their hive. And so, yeah, I don't like to do this, but I don't like losing colonies of bees from mites. So doing this is something that that it's necessary. There are variations. Some people use ether, like you might get from starting fluid. Others use soapy water, windshield washing solution, or even powdered sugar. But sometimes you can get misleading counts. And like the powdered sugar, it has to be done correctly. They have to be shaked for a few minutes, rested for a minute, and then poured carefully. If you don't do that timing correctly, you might underestimate the number of mites that are in your colony. The powdered sugar one, you're not actually killing the bees. So you could pour them back into the colony if you wish. But yeah, some of them will be pretty pissed off after you do that. And here's a little short video kind of just showing you my process for doing a mite wash with alcohol. I find a brood comb, make sure that the queen is not on that. I shake those bees into a bucket. The older bees will fly off, but the nurse bees, those are the ones that you want to use. Those tend not to fly, and you can get a half cup and put it into your jar. I use a mason jar, wide mouth, and there's a little screen that I've put on there. And I put the bees in there, and then I put some alcohol in there. You can use rubbing alcohol. Enough to cover the bees, and then I shake it vigorously to make sure they're all covered, and I let the rest of the bees go. After shaking vigorously for a bit of time to make sure all the mites, any mites that may be on them, detach, I end up pouring it off into a clear white bucket so you can see the mites, and I shake vigorously so that all the mites fall off. You can actually reuse the alcohol solution a few times before it gets too dark and yellow and has too many other particles in it. And then, after I'm done shaking it off, I look closely, and you'll see bits and particles of things that are not mites, but you will see mites that have washed off. There's a stinger floating around there. There's some other particle. But you can see quite a few mites in this. And if you have 300 bees, 3 mites would be a 1% infestation. And as you can see, this infestation is a lot higher than that. So basically, in order to figure out that percent, you divide the number of mites by 300 multiplied by 100, and you get the percent mites. So in that bucket where there were 9 mites out of 300 bees, that would be a 3% infestation. Now, some of us have large apiaries, and we can't go through and sample every single hive. So what you might do is, maybe sample 8 or 10 colonies in each apiary, or maybe 10% of an apiary, or something like that. To be sure, if there's any colonies I might be considering to use as breeding stock, I'll definitely be sampling them. The ones with lower percent infestation, all things being equal, are ones that I would be more likely to select as part of my breeding program, as long as they have all the other traits that I'm breeding for. And this is just showing, depending on how many colonies you have in your bee yard. And this is for larger beekeepers, the sideliners or commercial beekeepers. So, if you don't have a big problem with mite populations, usually you do colony testing about twice a year. But if the populations are high, you might, especially if you're in a warmer climate, you might need to do it every 2 months. Anytime a colony is not doing well, it's wise to sample, because Varroa mites are one of the top reasons for them to not do well, but not the only cause. There's other reasons for colonies to fail, nosema, queen failure, and other things. It's also important to think about where the mite population may be, at certain times of the year. You're going to have a higher mite population, in the fall and summer, and then the question is, when should you treat, and what timing should you treat? So let's talk about, treating for mites. Some of the factors that influence mite population, I mentioned time of year. You're going to have a higher population of mites at the end of the summer. How big the colony is there any hygienic behavior or Varroa-specific hygiene in those bees? How much brood is present? How much drone brood is present? And so on. And so the mite drop, if you're looking at the mite drop, will change during the season, and it might not be very much earlier in the summer, but it can really start to drop off a lot, especially as the brood production is shutting down. There can also be differences year to year. Some years are high mite years, other years might be lower. Maybe in a low mite year you might not need to treat as often. So this is a common question. What percentage of mite infestation is considered harmful and should be addressed? And this has been debated, by beekeepers, and it probably depends where you are, what time of year it is, and so on. And this is a really nice slide from the Honeybee Coalition, looking at what the percent is of the mite wash where you might be in the danger zone. And so if you're 1% or less, that's a really good number. If you're in the 1 to 3%, depending on what time of year it is, that's something that you need to be aware of, and you might need to be thinking. But once you get above that, 2-3% or more, then that's a real problem, and you better think about treating right away. Otherwise, colony loss may be imminent. And after treating, you really should have less than 3% on a mite wash, ideally even less than that. And this is just looking, and this is from Randy Oliver. He's got a great website. I cannot speak highly enough of the work that he's been doing, and not just with Varroa mites and treatments, but with all the other things that he is doing. But basically, you try to think about doing treatments before the mite population gets out of hand and the bees are damaged. So you're always trying to think about doing it before. And where I am in western South Dakota, our winter bees begin being raised around mid-August. So one common mistake that I see with beginning beekeepers is they do treat for mites, but they do it, middle of October, and you've kind of missed that ship has sailed, and so those bees are already damaged. And sure, better late than never, and you may kill a lot of mites actually, but then the colony still dies, and they wonder, well, I treated for mites. What happened? It's like you waited too long. Now, some treatments, formic acid cannot be used when the weather is warm. So we're really looking for a window, where we can do that treatment. But I try to get all my treatments done, by mid-September at the latest. You should always treat all colonies in a location at the same time with the same treatment. If you have a couple of colonies with high mite numbers, they might act as what we call mite bombs. Although perhaps we should not call them mite bombs, but maybe robber lures. The debate was, as these colonies are collapsing and dying, do the bees carrying mites move to other colonies, or alternatively, do other bees rob out those colonies and then bring the mites back to their own colony? And at least in this study that was done, it looks like it's the robber lure. So the colony that is collapsing is getting robbed out, and then those mites are brought back to the colony that was doing the robbing. So what are some of our current treatment options? We have many options available, and not all of them, require miticide or chemicals. There are controls that we call IPM, Integrated Pest Management. Some of them are more effective than others. Drone brood removal. It is a little time consuming, but you can remove the drone. You can get these green drone brood frames from Pureco and put them in the freezer for 24 hours, freeze the brood, take them out, let them defrost, and then put them back in the hive, and you will kill off a lot of mites that way. Some folks do brood breaks or brood interruption, or even requeening with more mite-resistant stock. Other treatments really haven't been shown to be that effective. Screen bottom boards were pretty popular a few years back, but that they don't really control or decrease mite population at all. Some folks were treating with powdered sugar, but that wasn't really doing very much either. Some of the first treatments that we had available, Coumaphos and fluvalinate, these were the first mite treatments available, but they really had a lot of disadvantages. They would get into the beeswax long term. They had a very long half-life, and now even though they are available through many bee suppliers, they're no longer recommended because they're not effective, and also some of the other disadvantages. So another treatment is Apivar. The generic name is Amitraz. It is a miticide. You can't use it when honey is available, and it's a slow-acting treatment, so you have to have it on for several weeks. There have been concerns about mites beginning to develop resistance to Amitraz. I've heard of some beekeepers using a double dose, which I really, hesitate to even, to consider that, because none of these treatments are non-toxic, so I worry about any harm that might cause, and it's not really labeled for that use. So unless there's research showing that using a higher dose is acceptable, I probably would avoid doing that. Then there are some of what we call the concentrated essential oil, volatile oil treatments. There's Apilife VAR, which has thymol, menthol, and some others, and then Apigard, which is solely thymol. The disadvantage is you have to go back every 7 to 10 days for 3 applications, but the benefit, it is naturally derived, and you have to make sure that, like a lot of the volatile chemicals, that the temperatures are acceptable and not too high. Then there's HopGuard. It's an acid derived from the beta acids of hops. It's put in the hive for 30 days. There have been mixed reports about the effectiveness of this treatment on mites, so I have not used it myself. Perhaps if there's more information showing how it is useful, I may consider using it. But because of the mixed results, I haven't used this in my colonies as of yet. And then we have the formic acid-based treatments. Formic acid is a volatile acid, and it is actually naturally occurring in beehives. Other species of the Hymenoptera, ants, will secrete formic acid. If you go to an ant hill and you pick up an ant and then you smell that ant, you might smell something pungent, and that's the formic acid that the ant is using as a defense mechanism. So it is very effective, but the temperatures have to be not too high, otherwise you might have queen loss or excessive brood loss. It is the only treatment that is effective in capped brood, and it's also okay to use when there's a honey flow. You can use either 2 strips or 1 strip applied twice. There's actually 2 strips. I put 14 days. That's actually not accurate. It's 2 strips over a week. Or 1 strip applied for 10 days, applied twice. Then there's oxalic acid. This has been used for many years in Europe. It can be used as a dribble or spray, sublimation, or what some people call vaping or vaporization. There also have been some that have been experimenting with oxalic acid in pads. And I really don't cover any of that here, mainly because Rennie Oliver has done so much work with this. I recommend that you go to his site and you read what he has written and you watch some of his videos and presentations. He's really done so much work looking at trying to use oxalic acid in like a pad. Hopefully, it will work out, so we have an alternative to some of the other options, and hopefully it'll work out in a way that is not expensive. Some of these treatments are more expensive than others, and oxalic acid is probably the least expensive. It does not work very well when brood is present, and you have to be very careful because it could be potentially harmful, so you have to use the proper safety equipment. So what is oxalic acid? It's a naturally occurring acid that occurs in vegetables, rhubarb, spinach, and some others. And 1 pound of carrots has roughly the same amount of oxalic acid used to treat 1 hive. Although our treatment recommendations are starting to evolve, but I'll cover that here in a second. So for those of you that don't want to invest in the gadget to do vaporization, you can use an oxalic acid dribble. This is from Randy Oliver's site. And dribble it over the brood and over the bees themselves. In places that have hard water, such as what I have, because oxalic acid is an acid, it may react with that, and so it's probably best to use distilled water. They dissolve more readily in hot water, so you may put a small solution in hot water first, and then put it into the sugar solution after the crystals have already dissolved in the hot water. So you dissolve it in very hot water before you add to the sugar, and once it's fully dissolved, then you can add in the sugar. And here's again a photo from Randy's site. You dribble in about 5 milliliter per seam of bees, and you got to put it directly on the bee's body. So if there's no bees on a certain frame or comb, there's no point in dribbling any oxalic acid there. It is most effective when colonies are broodless, so that would be in the middle of the winter. Or it could be if you've made a split and the new queen has mated. Let's say you did a walkaway split, or maybe you put a ripe queen cell in there. You could also use it during that time also, because she might begin laying eggs, but there's no capped brood, between around day 21 or so after you've made that split. But otherwise, in full-size colonies, this is a most effective cleanup treatment in the fall or winter when there's very minimal brood present. Many of us, including myself, use oxalic acid vaporization. I like to think of it as being gentle on the bees. It doesn't really cause harm on the bees or the brood, but if you don't take precautions, it could be potentially hazardous if it got in your eyes or you breathed it in. You could use it in cooler weather. Now, I wouldn't do it when the bees are tightly clustered and it's below freezing, but in temperatures that the bees are active and they're moving around inside the hive, even if they're not really flying, you can use that. Previously, we would recommend 2 grams per 2 deep box hive, but it's come to we've learned through research that dose may be too low. So perhaps we need to be using a higher dose, and I'll get to that here in a moment. Some beekeepers would recommend, to get away or get around the fact that oxalic acid does not treat mites in the capped brood, treating every few days, every 3 days, every 5 days, for several treatments. And so this study was just published in 2021, looking at 7 applications of oxalic acid vaporization 5 days apart. And basically it showed no benefit. It didn't cause any harm, but it didn't actually lower Varroa mites in any significant way. Now, one point I have to make is they use the standard treatment, 1 gram per deep brood box. That is what oxalic acid is labeled to be used as. And the question is, now that we're learning about higher doses might be needed, would this treatment, this every 5 days treatment, been more effective if they used a higher dose? The simple answer is we don't know. Somebody will probably need to repeat this study, with doing repeated treatments every few days, but using a higher dose. This study was also just published very recently. They looked at 40 colonies, 10 in each group, and they looked at different doses. 1 gram of oxalic acid, 2 grams, 4 grams, or none. The hives that got none obviously didn't do very well. And these treatments were applied via vaporization once per week. And they looked at the differences. And they found that those that received 4 grams showed significantly lower infestation rates, with applications of 2 to 4 grams of oxalic acid significantly decreasing mite load. So that means that perhaps if we're going to be treating, whether it's one time during a broodless period or multiple times every few days, that perhaps we need to be using higher doses. And I have to admit, I've noticed this myself. When I was using the 1 gram recommended dose, and I would do another mite wash afterwards, there were still quite a few mites present. And so I myself have begun increasing the dose. I didn't really tell very many people about that because I didn't have any science to support that approach. But now it's nice to have some science to show that, yes, probably we need to be using higher doses of oxalic acid when treating our colonies via vaporization. Some of the other options that have been used, some folks in Europe cage the queen for a period of time, then they release her and treat about 4 or 5 days later. This is enough time to allow any brood within the colony to emerge, and so there will be no capped brood, and so the oxalic acid could be treated. The disadvantage, though, is there can be supersedure. It's not really a good thing for a queen to be caged that long, and the colony of bees thinks something is wrong with the queen, that she's not producing brood, and so they often will try to supersede her. Requeening with a new queen, so caging the old queen, remove, basically killing her, and then put a new queen in, treating 5 days later. Requeening with a ripe queen cell, so that queen cell will need to emerge, go on mating flights, begin laying eggs, and so if you treat between around 19 to 20 days later, there will be brood in the hive, but none of it will be capped. If you do walk-away splits, treating at around 20 days for the same reason, the queen has to emerge and then and then mate, and so at 20 days, all of the capped brood will have emerged. Package bees or swarms, you can treat at day 5 before day 8, before there's any capped brood present. So one single treatment of oxalic dribble or vaporization can be very effective in that situation also. And so comparing the dribble versus the sublimation or the vaporization, the dribble is effective, it's safe. You still have to wear gloves and eye protection, but you don't need any fancy vaporization equipment that's expensive. The downside is you do have to open up the whole hive, and it may not work quite as well if weather is colder. The vaporization or sublimation is very effective. You can do it without opening the hive. You can do it in colder bees, and it appears to be gentler on the bees, but you do have to have a specialized equipment that is expensive. You have to not be very careful not to breathe in the mist. And some of the pan types that heat up, if they touch anything flammable, they could start a fire. There have been situations where beekeepers set the entire hive on fire because it touched the wax. And if, wax is flammable, and once a wax fire starts, good luck putting that out. Now, there have been many other treatments that, people have come up with over the years, and we don't recommend it. Mineral oil fogging, oxalic acid and ethanol, my gosh, that's potentially flammable. Powdered dusting is okay for sampling for mites, but it doesn't really lower mite population. A lot of us use screen wire bottom boards, but they really haven't. I mean, it's nice to see those mites die after I do a treatment, but you really aren't controlling the mites by using those. They've tried thermal treatments. Yeah, they can lower the mite counts, but you will have brood loss. And the question is, how practical is this, in a real life situation, or how effective? Some folks were dabbling in small cells. They had the idea that Africanized bees had small cells, and if the bees are smaller, but it doesn't work. They've done studies looking at this. And then mite traps, trying to trap or catch the mites, they just haven't been shown to work. This next one, when I first saw this, I thought this was a joke, the bee gym. It turns out it was like actually being marketed. The idea the bees would go and scrape the mites off. We have to remember, bees do what they do, and just because a human or maybe a dog or another animal might try to scrape off a parasite, that doesn't mean that's what the bees are going to do. I've never actually seen a bee in my observation hive with a mite trying to go around and scrape it off, scrape the mite off on anything. So I thought this was a joke, but this is actually a commercial. This was actually sold as something to put in the hive. As a beekeeper who's been keeping bees for 4 decades, it seems like every decade there's a new gimmick, that they're trying to make money off of, and trying to get money from the beekeepers. So I recommend using standardized treatments that we have been proven to work. So to come to a conclusion of this presentation, all colonies, if you live in the U.S., North America, all colonies already have mites or will have mites. And just because you don't see them doesn't mean they're not there. And if you're not sure, it's always wise to do a mite count. Once the population starts going over 3%, you're going to start losing colonies. And so in order to avoid that from happening, we're trying to keep mite counts low through treatments, through integrated pest management. If you have colonies that, are full of mites, this can result in the mites spreading to other colonies in your bee yard. And we are trying to breed for mite-resistant hives, bees, and although that definitely helps in reducing how fast the mite population increases, we still aren't quite there yet in being able to go completely treatment-free, unless, of course, you're willing to lose lots of colonies by doing that. So this is a summary of treatments. I looked at the cost of these quite a few years ago, and I know prices go up and down, so please don't quote me on this. These prices may not be accurate, but they are useful to know. Some of these can be more expensive or less expensive. And comparing these, the Apivar, the Amitraz, as well as oxalic acid, can be used at practically any temperature. Not under freezing, not when the bees are in their winter cluster. Probably that's not going to be able to work as well. The oxalic acid vaporization may not get in there. But pretty much it can be used at any temperature, if the bees are active and moving around. There are a few treatments that are okay to use when honey supers are on. Oxalic acid was recently approved for that purpose. Other treatments cannot be used, and that includes the thymol-based treatments as well as the Amitraz. So you need to keep that in mind if you need to do a treatment during the honey flow. The least expensive treatment is oxalic acid dribble. The treatments with the vaporization are also not expensive, but you do have to invest in some type of equipment to allow you to treat with that method. Amitraz is not considered an organic treatment, but these other ones listed are within the organic or naturally derived ingredients, whereas Amitraz is a synthetic. These all can work, and they all have their place. So the way that I think about it is, I do a mite sample in the spring to see how the colonies have done, and if the sample is pretty low, then I will not do a spring treatment. If it is higher than that, though, then I would treat prior to the honey flow, so I don't have to worry about it during the summer. And if I resample, did the treatment work, then great, we're good going into the honey flow. And then after the honey flow is done, but before winter bees, as winter bees are beginning to be raised, I will do another sample to see. The percentages will be higher towards the end of the summer, but as long as it's less than 3%, we're okay. If I'm going to do one treatment a year, it's probably going to be that one midsummer. I've just been caught off guard too many times over the years and lost colonies over it. And then as I'm preparing my colonies in the winter, wrapping them, I might consider an oxalic acid treatment as a cleanup to clean up any mites that I might have been missed by the late summer treatment. And it's really important to try to rotate treatments if you can. Try not to use the same treatment over and over again, so that we can minimize getting resistance. As far as we know, there's not been any resistance to formic acid or oxalic acid, but we have been seeing possibly some resistance to amitraz, which is of concern. So that brings my conclusion to the end. I hope this information has been helpful to you and will help you avoid losing colonies to these mites. It's really one of the most frustrating things about beekeeping now. It's something we didn't have to worry about back in the day. If you're going to keep bees successful, you need to have a plan for how you're going to manage the mites, whether it involves treatments or IPM or a combination of both. Thank you so much for watching my presentations. Have a great day.